There are a lot of different cancers, and the cancer we are focusing on is our brain cancer, and more specifically glioma. Glioma may be quite benign disease or very, very malignant disease. Unfortunately, the most common form is glioblastoma, also called grade 4 glioma. How long patients survive to this terrible disease is quite variable, but the median survival is only 15 months. Certainly now the combination of surgery, radiation and temozolomide or chemotherapy, in more global words, does improve a little bit the prognosis of patients suffering with this glioblastoma. But it's really modest. And most of the patients do not benefit really, but a fraction of patients benefit quite a lot. 10 to 15% of patients may survive several years due to this combined strategy. The trial, which is now supported by the Gateway for Cancer Research, is a trial of therapeutic vaccines. In a few sentences, we are now convinced that our immune system is able to recognize and, in ideal situation, to destroy tumor cells. It's very difficult to build a vaccine because we have to have as compound which are specifically expressed by tumor cells and not by normal cells. And it is not so easy, because to understand that you have to realize that each cell of our body express at its surface around 50,000 different antigens. All the compounds of the cells are expressed like that. And the immune system tolerates normal cells because it is educated for that. Our immune system is educated to not attack our body, to try to avoid autoimmunity or autoimmune disease. When a cell is becoming a tumoral cell, some, but only a very few of the surface compound could be different. This is what we call tumor antigens. So the goal of our research was to identify a very few. At the beginning we hope for one, but to identify a very few antigens at the cell surface, because only a very few antigens or cell surface compounds are different from tumor cells and normal cells. And this is what we succeeded last year, after more than 15 years of research with this goal, and we finally identified 10 antigens which can be considered as 10 targets to which we have now to educate the immune system, and mainly T lymphocytes, which are the effector cells able to recognize and to kill tumor cells. So we have to educate these T lymphocytes to target the tumor cells, and hopefully, because these targets are not or very poorly expressed by normal cells, we hope that we can avoid collateral damage to the normal brain, to the brain. Yeah. To develop a new drug, usually you need 5 to 10 years. Here, clearly, we have to develop a new concept of treatment. So we also anticipate a 5 to 10 years period of research, of clinical research now, at least, to see how much this type of treatment, immunotherapy, may impact the outcome of patients suffering from this cancer. I really thank all the donors. Your support is just amazing for us, and for this critical step, we really believe that this clinical trial is a very crucial step for the further development of vaccine, not only at Geneva, but around the world, because identifying glioma antigens and now doing the first trial will certainly expand the clinical development of all types of immunotherapy against brain tumors. This could be the step that could, this hopefully in the future would be designed as very important for hopefully the success of immunotherapies in the treatment of brain tumor patients.